So our final presentation is from Laura Ganser. Laura is a third-year graduate student in biochemistry. She's in the laboratory of Dr. Al-Hashimi, and her research interests focus on the structure and function of RNA and focused on developing strategies to target RNA with small molecules for HIV treatment. And her presentation today is structure-based methods to target HIV transactivation response element RNA.
Thank you. So as she said, I'm Laura Ganser. I work in Dr. Hashimi's lab in the biochemistry department. So I'll be talking to you about our efforts to target the HIV-1 TAR RNA element using structural and dynamics-based approaches. So HIV-1 TAR is a highly structured, highly conserved RNA in the 5' untranslated region of HIV. It acts at the transcriptional level by recruiting TAP and PTFB to the stalled RNA polymerase. So then the TAP-PTFB complex phosphorylates RNA polymerase as well as transcription factors and allows for elongation and transcriptional activation. So because of its function, it's been an attractive target for HIV therapy. Its role in transcription is completely essential for viral replication. Additionally, because it acts at transcription, it's an appealing target because there are currently no inhibitors for transcription. Additionally, acting at transcription could help address latency because by preventing low-level ongoing transcription in latent reservoirs, which could lead to a state of deep latency and possibly a functional cure. Finally, the structure and function of TAR are highly dependent on its sequence, and therefore it's not prone to mutate, which is always good in a target. So for all of those reasons, it's been targeted by several groups in the past. However, there have been very few successes based on the inherent challenges with targeting RNA. For instance, many of the available small molecule libraries are biased towards compounds that bind to proteins. Additionally, most RNA is found in canonical Watson-Crick mispairs and A-form helices, which this structural homogeneity leads to problems with specificity. Finally, just finding a compound to bind RNA in vitro does not necessarily mean that we'll get biological effects, partially due to the specificity problem, but also because RNA is prone to be bound by charged or highly polar compounds that have poor cell permeability, and also just maybe because they don't bind tight enough to induce a biological effect. So also, structure-guided approaches have failed because static structures fail to represent the flexibility of RNA molecules. So this is highlighted with HIV TAR. What I'm showing here in the center, we have free TAR. These are all high-resolution structures of TAR, either free in the center or bound to small molecules. And so you can see there's a wide variety of conformations that TAR takes on when it binds its compounds. So you can imagine without knowing a priori the structure, the bound structure of TAR, it would be difficult to use conventional docking methods. So my lab developed a method using an NMR technique called residual dipolar coupling, which gives experimental information on the structure, as well as molecular dynamic simulations to generate an ensemble of structures that better represent the dynamics of TAR in solution. So interestingly, this dynamic ensemble of free TAR includes structures that are highly similar to the solved structures of bound TAR in the PDB, supporting that it is in fact sampling correct positioning. So so our approach to docking uses this dynamic ensemble rather than a static structure. And to do this, we use a program called ICM, which scores ligand binding to specific receptors using the equation shown on the bottom, which is meant to mimic binding affinity. So what we do is we take each ligand and dock it to each of the 20 receptors and take the top score from that. And this method has been published and is validated by showing that we are able to get a high correlation from docking score to known binding affinities. So to further evaluate this approach, we did a parallel screen of over 100,000 compounds using our virtual screening approach and an experimental high-throughput screening approach. So for the high-throughput screen, we used a TAP peptide, which is the protein that binds to tar. And on either end of this peptide, we have a fluorophore. The peptide is highly flexible, so when it's free in solution, the two fluorophores interact and quench each other's fluorescence. When it's bound to tar, they're held at a distance so that fret can occur. So as we titrate in our inhibitor and displace TAT, we see a decrease in fluorescence, which is shown with my neomycin B control. We additionally did this in the presence of 100-fold excess tRNA to show specificity. So neomycin B is not specific for tar over 100-fold tRNA, so, which is why we see a decrease in the binding affinity. So the workflow for our high-throughput screen, first we did an N equals 1 with a large excess of compound for all 100,000 compounds. We then took the hits from that and did a confirmation assay where we did it in triplicate. Hits that were reproducible were subjected to dose response curves and tested for fluorescence intensity or for fluorescence interference. So finally, we were left with 17 compounds, but these we tested with NMR to ensure that they actually are binding to tar, and we found several false positives from the high-throughput screening. So finally, we were left with 7 hits. And these 7 hits actually fell into 2 families of compounds, the top of which is not selective in the presence of 100-fold excess tRNA, and the bottom are anthraquinone derivatives, which are in fact selective in the presence of tRNA. So because we did parallel screening, we also have all of the docking information on these compounds. So what I'm showing here are the predicted poses from docking. So these are the poses that gave the best scores from docking. And we see that the 3 compounds that are highly similar are all predicted to bind the same conformation of tar, and they're predicted to bind in a similar manner in the bulge. And so we were able to use NMR spectroscopy to show that in fact these compounds are inducing very similar changes in the tar spectra. And this supports not only that they are binding similarly, but they are binding similar to the manner predicted by docking. So if we look at the docking score for each of these compounds, we see that that docking does give them good scores. 4 of the 7 are in the top 10% of our DOC compounds, and the remaining 3 are within the top 50%. And it's important here to keep in mind that this is the primary screen, so we had very low sampling for these compounds in docking, but if you increase sampling time, we do see an enrichment of these compounds, or further enrichment of the true hit compounds. So using this information, we can make a ROC plot, which compares the selectivity versus the specificity. So in the ideal case, all of your true hits are selected before false hits, and you get an area under the curve of 1. In the random case, your true hits are distributed evenly throughout your false hits as well, and you get an AUC of 0.5. For this screen, we were able to get an area under the curve of 0.81, which is reasonable and is comparable to docking programs or docking campaigns against known structures. So we were also interested to look at the compounds that were highly favored by docking and why were they excluded. And actually, we found that 5 of the top 15 compounds from docking actually do bind to tar by NMR, and they were missed by high-throughput screening either due to experimental noise or because they don't bind tightly enough to displace TAT, or additionally that they bind but don't kick off TAT, possibly by binding to the apical loop region. So what this means is that actually our area under the curve of 0.8 is an underestimation because high-throughput screening failed to identify several compounds that do in fact bind TAR. So from these parallel screens, we summarize that docking to a dynamic ensemble is able to identify small molecules that bind TAR. Additionally, multiple screening approaches is very useful in finding false negatives and false positives. But despite the success of this, we only identified basically 2 classes of compounds, and even taking into account the other compounds we found, our hit rate was very low and only 0.01%, and none of these compounds had activity, had biological activity. So this begs for further approaches and further strategies to target RNA. So real quickly, I'm just going to go over our new direction for this project. So everything I've been talking about, the dynamic ensemble we made is for the ground state secondary structure of TAR. But in fact, using NMR relaxation dispersion techniques, we recently found that TAR will transiently morph into other secondary structures, which we term excited states. And so we propose that these excited states may be better targets than the ground state, first because they sequester residues necessary for activity, and it's well established that that trapping a conformation or a biologically inactive state will lead to better biological activity. And also because these excited states are rich in non-canonical motifs, which we predict will offer more unique binding sites, which may help the specificity problem. And so using a conformational model of our second excited state, we actually see the formation of deep and unique pockets that may be good for potential targeting. So finally, to see if actually trapping excited states would lead to activity, we used mutations to either trap excited state 1, excited state 2, or to knock out the excited states and trap the ground state conformations of TAR. And in an activity or a transcriptional activity assay in HeLa cells, we see that trapping the first excited state reduces transcriptional activation by greater than 50%, and trapping the second excited state nearly kills transcriptional activation, whereas mutations that preserve the ground state have recovered activity, including a rescue mutant for one of our ES2 mutants. And so finally, in vitro binding assays to TAT mimic these results, suggesting that our decrease in activity is due to the inability to form the TAR-TAT complex. And so this data overall suggests that if we are able to find a compound to trap the excited states, similarly to how these mutations do, we would be able to prevent transcriptional activation. And so our future directions are to determine a dynamic ensemble of these excited states and then to use a structure-guided approach to target them. So I would like to thank everyone in my lab, but especially my advisor, Dr. Hashim Al-Hashimi, as well as our collaborators, Dr. Brian Cullen and Hal Bogard, and Dr. Paul Viñas for the in vivo work. So with that, I will take any questions. Thank you.
Questions for Laura?
That was a nice talk, Laura, and emphasizing the importance of dynamics in a lot of these functional states. One question, though, relative to the hits you got, and particularly the hits you got that weren't revealed by screening. Do you have any idea what the range of binding constants was for those?
No, so we were able to, so for one of them, We do, because when we repeated the assay, it did have activity, so that one was just lost in the noise. But for the other ones, we don't, because for some reason they aren't active in the assay. And so we would have to do a full NMR titration to get the KD, but as of yet, I have not done that.
Other questions?
When you, it's a very nice talk.
Thank you.
When you carry out your simulations, I guess, do you ever account for the fact that you might have protonated base pairing that might be involved in some of this?
In the RNA or
in the
protonation in the?
Well, in your targets or in the actual.
So that's something that we would have to include beforehand. So typically we don't. However, if you saw like in the first excited state, there is a protonated AC mispair. So for that, if we were to dock to that, we would need to include the protonation. The way that docking works is you have your structure, and that's pretty static, whereas your ligand can come in and be more flexible, and that's where you can kind of mess with charges and things like that. So no, it doesn't take into account changes in protonation.
But you could evolve a model in which that was the case, like
if we had any reason to
protonated
believe,
CC or things like that. Because do you have any? Well, we can talk about this later.
Okay.
Thank you.
Okay, great. And then maybe one question, we heard the previous talk about the problem of resistance to ARVs. What do you think the outlook would be for resistance with an RNA targeting approach to treatment?
So that's one of the benefits of TAR, is that it's so highly conserved, and it really does not tolerate very many mutations. So in that sense, it would be good. And also, if you think about like art therapy now, you target many different stages in the viral life cycle. And so if we could target the TAR-TAT interaction, we would be adding another step that we could be targeting, which kind of may have a collaborative effect, maybe to help.
Great.
Thanks.
All right, thank you,
Thank you.
Laura.